Good morning, everyone. I'm really delighted with the speakers that we have today. Last spring, we were getting a lot of comments that we wanted more in-depth, climate-oriented talks. So what we're covering today, we're very pleased to introduce Dr. Michael Kent and Dr. Corbin Schuster from Oregon State University. Michael Kent is a valued member of the Department of Microbiology and Carlson College of Veterinary Medicine at OSU, and Dr. Corbin Schuster is a microbiologist and fish disease expert. He's a member of the Yakama Nation and is active in engaging these important stakeholders in collecting samples from the field and studying the results, as well as to identify ways in which the community can be engaged. Because as I've said before, only by banding together can we help each other and improve the world. So I'm very delighted today to welcome Dr. Michael Kent, Corbin Schuster.
Well, thank you, Anne, and the organizers for having the opportunity for Corbin and I to come up here and talk with you folks. We met as I guess it was an Easter brunch at the vineyard, the Willamette Vineyard, and that started the conversation. And here I am now. The vast majority of my speaking is in teaching classes and going to conferences and all this. And I realize now that this type of venue is really important, is sharing information with people outside of our academic homes. I think it's really very important. So I'm going to talk about prespawn mortality in Chinook salmon in Oregon today, specifically. Is that adequate? Okay. I'm going to be talking about prespawn mortality in Chinook salmon, specifically in spring Chinook salmon, and specifically focused on Oregon, but we're going to go beyond that a little bit. So I don't have time to go through all the details of our collaborators and then even our funding agencies. We've been working on this since 2009, and people have come and gone as collaborators. Some of us are still been working on this consistently. You can see Corbin down near the bottom. He did his work and PhD at Oregon State University and his postdoc working on zebrafish, and now he's joined this team. In fact, as you can see, I'm emeritus. I'm on the way out. I'm turn the project over to Corbin. He's now the lead on this work. And I just added this in now. This is a very dynamic program that we have going. Now we've added a virologist, Tom Walczyk from Washington State University, with some exciting findings he has found in our fish just recently in the last several months. And acknowledgments to our funding agencies. Again, because limitations of time, I can't go through all of them. And I probably don't have every one of them on there. You can see tribes on there, the Army Corps of Engineers, Washington State University, Oregon's Department of Fish and Wildlife, Heritage University, and others. And these are the main contributors, both financially and as collaborators. So we're going to divide our talk today. We have 2 hours. And so with breaks, we're going to take advantage of this and break it up into 4 sessions. One is the background on on Chinook salmon and prespawn mortality. Then I'm going to talk briefly about methods and science and how we use the methods that we use for our investigations of diseases in wild fish and impacts of diseases and pathogens in wild fish, and in particularly this situation here. Corbin's going to give a short talk on community outreach and engagement, and then I'll finish up with really kind of more of a nitty-gritty talk that I would give to colleagues at a conference on the recent discoveries that are underpinning what we feel are very significant contributing to prespawn mortality, called a disease called adult salmon enteritis, and the etiology of it as well too. And the way I have that set up, it seems like I'm going to be talking about a lot. I will gauge as we're going how much into detail, how little or how much we get into detail on the etiology of the disease based on time. Of course, I'm going to give you the overall picture of it, but we'll have time to get into, we'll see how much we can get into the details in our second hour. So we're going to talk about background, general physiology of adult salmon, a little bit on Pacific salmon life history. I see a lot of people here already know salmon pretty well, so I think I'll probably be able to go through this pretty quickly. Then, as I said, methods for investigation of diseases and mortality in wild fishes. Then Corbin will give a talk about citizen scientists and Native American stakeholders. And then I will finish up on the etiology of this phenomenon that we're seeing. So salmon are important, particularly Chinook salmon. We see economic, tribal, and ecological reasons. And there are others as well, too. So these are iconic species that we're very interested in. And so now, really quickly, many of you are aware of this, we're going to be talking about Oncorhynchus species in general. That's the Pacific salmon, including rainbow trout. Rainbow trout and steelhead are in the genus Oncorhynchus. In general, we see this, they spawn in early development in freshwater with variations, depends on the species, and then grow out as adults in the ocean. They're simulparous. That means they die after spawning. It's like a flower that, an annual plant that's going to die after it reproduces. And that's very key to this thing. So we talk about pre-spawn mortality. These fish are destined to die anyway, but where the problem is, they don't make it to spawn. So instead of they're going to spawn in the fall and then die afterwards, what's happening in pre-spawn mortality, they may die in August or even sooner, like in July, and it's very high numbers. And that's what the big concern that we have, at least. So where we're going to be talking about today, try to steep on this, is this phase in the life cycle where they come back into fresh water and die before spawning. So this is the events. This is a part of the life cycle that we're going to be focusing on today. Again, pre-spawn mortality, this is another way of looking at the development. We see they go through the whole life cycle in freshwater, juveniles, and they go into the ocean, then they come back. And then they come back for spring Chinook. It would be winter or spring where they come back and then stay in the river over summer, and then they spawn. But then what happens is pre-spawn mortality, they die too soon. So Chinook salmon. So here are the other species. I'm going to focus on Chinook. So as I said, I'm not going to go through each one of our different Oncorhynchus species. Each one has a different strategy on life cycles. Like sockeye have a lake development in freshwater, longer in freshwater. Amongst the Oncorhynchus species, steelhead are the only ones that are not strictly simulparous. Some of them will survive after spawning. But let's talk about Chinook. That's what we're talking about today. Spring Chinook. They come back as adults winter or spring into rivers and spawn in the late fall. So they have to survive that long period, let's say 6 months, in fresh water before they spawn. Versus fall Chinook, which return late in the fall and spring and spawn shortly thereafter. Here's the area that we're talking about, where most of our work has been done in the Willamette Basin. The Willamette Basin, as many of you are aware, it's centered around the Willamette River and tributaries. We see spring Chinook are native to upstream of the Willamette Falls, which I'm showing here near you guys. They spawn in tributaries, and then they're listed as ESA threatened species, so environmental sensitive units. So these are listed as important species. A little bit, I'm not going to spend a whole lot on the geography, but just kind of the broad brush of what we're looking at. Very simplistically, we have the Willamette River that runs north into the Columbia River. Then we have a number of tributaries where the fish will ultimately spawn. But along the way, many of these spawning locations, we have hatchery traps, major dams. So they have to get above these traps and dams to ultimately spawn in the upper reaches. And this is where we have a lot of problems. It's not just a straight shot up to their spawning grounds in many situations. So upper Willamette, they're a listed population. Again, this is just a map showing again. So this is risk of extinction. You could see very high for many of these sub-runs, if you'd say, off the Willamette system. Okay, so we start out with the Willamette Falls. How do they get past that? Many of you are aware that we don't have to, there's a fish ladder. So we don't have to actively move the fish above the falls. We have a fish ladder. So but that's just the first obstacle in their journey. Many of the other times to get to their spawning place, they have to be trapped and hauled. So they're catching these fish, these adult fish, large fish, trying to get upstream. They can't make it across the barrier. So they actually have to be collected, put in trucks, and then hauled up above the particular barrier, the particular dam, and then released above the dam. Quite stressful for the fish. So a little bit about the fish. What are they enduring? So this is their journey. They come back. Remember, they're semmelparous. They're not going to live. So they stop feeding when they get to fresh water. They're exposed to a variety of freshwater pathogens that they had not experienced for a long time being out in the ocean. Also, they may have some cryptic, low-level infections of pathogens that they became infected with as par before they smolted. They may still be carrying those. Other stressors, as I said, big one is getting past the dams. So they're immune compromised also. So they're not feeding. They're profoundly immune compromised. And there's been lots of discussion, why is this to the advantage of all the fish to have the cortisol levels? You know, glucocorticoids are a response to stress, right? And then we always hear about how bad stress is and how cortisol is bad, etc. Doesn't matter if these fish are stressed or not. They're going to have high levels of cortisol. It will even go higher if they're stressed. But they intuitively have all that. And we can talk, when we get a chance for the discussion, we can talk a little bit about what are the advantage. Why would this be an advantage to these semmelparous fish to have very high cortisol levels at the end of their journey? So this is because we're going to be talking about infectious diseases. So an underlying concern is these fish are profoundly immune compromised. That means they're going to, as I said, underlying infections that they had as par may take off, etc. So initial benefits of this cortisol level is anti-inflammatory. You know, so you have an inflammation situation where the doctor wants to tamp that down, and they'll give you steroids, and that's what these fish are doing. So that's one thought, is that you're tamping down the tissue reaction to all these things. However, the negative part is increased disease susceptibility when you have these steroids. We see microbiome dysbiosis. I don't know if you guys have been with your group have heard microbiomes and diet are a big topic now. We're doing work on microbiome in these fish as well too, showing that there's microbiome dysbiosis. There's energy depletion, decreased reproductive fitness. This is in general considered with excessive cortisol. And then it could be death, prespawn mortality. So here's another way of looking at it chronologically. This is the normal scenario. The fish come into freshwater. They are going to experience exposure to freshwater pathogens. As the summer goes along, depends on the river, the time of the year, all sorts of different variables. But in general, they often are susceptible to, are exposed to, challenged by increased water temperature. But they survive all this. They make it till fall. They spawn and then they die. That's what we hope to see happen. But this is what's happening with pre-spawn mortality. Everything is accelerated. Everything is coming too early, including death. So they can make it all, they're subjected to all the same stuff, but then they die before they spawn, and that's bad. And it can be, I'm getting ahead of myself, you can have runs well over 50%, is not unusual. And there's runs where there's been 80 or 90% of the fish die in a given run. And this is kind of, I'm leading into what we're going to be talking about more in the second hour. Along with this is increased pathogen loads. This is a generalized graph that we've done 10 years ago, but it still applies, is a variety of pathogens overall increasing in the fish until the time they spawn. I'll show you a few maps here. I'm not going to spend a lot of time on these, just kind of getting a little more granular on that big map of challenges that you see where there's having to have trap and haul situations, and they're usually associated with dams. You've got to get them over the dam, etc. Here's one where we do like for example, at Cougar, Mackenzie hatchery. We have this, we've got to get them above these areas. So you see this throughout the Willamette system, this paradigm shown over and over again, where you got to trap and haul. You got to get the Willamette Falls, they get over fine. But then those other secondary things, man has to intervene and get in there and move them across. So, the purpose of the talk today is pre-spawn mortality, right? And so what it is I kind of break it down into a couple of 2 or 3 areas. We talk about extrinsic or abiotic roles. Ocean, so we've been talking about challenges in freshwater. Let's go back. We go in the ocean. They could have a poor ocean condition. Remember, these fish stop eating when they come back, right? So they have to be at optimal fitness as far as their energy levels, their fat storages, etc. If food is not plentiful in the ocean, let's say temperatures are too warm. That could be one reason. Currents, temperatures too warm. They may be not in the top fitness when they come back in from ocean. So ocean conditions, what are we going to do about that? That's a tough challenge. That's a global challenge, right? Water flow and temperature, oxygen. So this is more regional. I see that they put on my talk climate change. That's the big picture. But then we can have much more regional changes of effects of temperature by removal of riparian situation around the rivers, or changes in flow, putting dams in, taking dams out, etc. So changes in flow and temperature. That's kind of more year to year, seasonal. So we'll see wet years and dry years. So yes, we see this generalized trend of climate change, but you're going to have good years and bad years bumping up and down throughout that. I'm going to show you an example of that in one of my graphs. How about hatchery versus wild origin? I'm going to talk a little bit about a paper one of our colleagues, Jim Peterson, published a few years ago. He's an excellent fisheries biologist and mathematician, modeler, showing links. What is the situation with if hatchery origin fish versus wild origin fish as it relates to pre-spawn mortality? I'll talk a little bit more about that later. Then I've already talked about the troubles with dams. What about intrinsic? Time of returning to fresh water. So they've got to survive this whole time in fresh water. Historically, and they've changed this policy now, you think about hatchery management. You have all these fish coming up and we go, we need 200 broodstock fish. So they inadvertently would take, okay, we got 200 great fish, done. We got our 200 fish for next year. So they would lean towards collecting early run fish. The fish have evolved to cover their bases. Some come back early, some come back late to cover the bases for survival. And then if we intervene and take the fish, only the early returners, you're going to genetically down the road, you're going to be selecting for fish to come back, or you're selecting for earlier returning fish. That's been a problem, but I don't want to blame that. That's a historical problem, and now they're addressing that. So returning time to fresh water, response to stressors. Are they the poor ocean conditions, other things, genetics, all sorts of other things, underlying pathogens? How are they just responding to the stressors that they're going to have anyway, the stressful journey to get up to spawn? And then this is where Corbin and I work on diseases. We bring this is a whole other aspect that we'll be talking about, particularly in the second hour. So these are my former students. I guess she's my student still. She's just finishing up her PhD, even though she's gone off to her job in Wyoming. You'll see her picture. She did the work on the transmission stuff. Tamsin Pauley, her side thing is she's an artist, so I want to give her credit for these AI artistic images that she did. So this is, showing what a slide she put together. This is our normal life cycle, and then here's our pre-spawn mortality situation. So I asked her, I said, did you draw all that and stuff? She said, no, I went to AI and said, I want a picture of a dead salmon. and then they got in the water, and so then she selected it and took it from there. So here's some real pictures. This could be post-spawn fish, and that's fine. That's what the river is supposed to look like after they've spawned. And those fish can provide important nutrients in the river for the next generation. But if you see this in August before they've spawned, that's when we're going to see pre-spawn mortality. On top of this, I'm going to talk. We've seen this general decline in the total counts of fish coming back. So you have pre-spawn mortality, and you've got fewer fish coming back. And we'll talk a little bit about an interesting link to this. There's a little bit of buffer that's in there, fortunately. You can't beat down the fish as well as we're trying to, it seems like. ODF&W, this is a busy slide, but I want to give them credit. Our colleagues, ODF&W, Luke Whitman and others, they have provided lots of the samples for us in early years when we were doing this project, and also the data on levels of pre-spawn mortality. It's a lot of work, going out in the field through the summertime and looking at carcasses and evaluating them, opening them up and seeing if they spawn or not. They can do that with the females. You just look in there and see how it's full of eggs. It's a pre-spawn mortality. Males are more difficult because a post-spawn male is still going to have a lot of sperm in its testes. So we do, for males, we basically have a cutoff date, is if they die before September 1st in the Willamette system. Other systems would be earlier. It's arbitrary. A dead male in August, we're going to call that a pre-spawn mortality because they don't spawn till in most of the tributaries, they don't start spawning till after mid-September. So here's some data just showing you pre-spawn mortality. This is on the Mackenzie River across the years. You can see the lower Mackenzie, was much higher than the upper Mackenzie. So if they get up to the areas, they were able to spawn. This is quite a shocking slide. If you look at it, what we're talking about is pre-spawn mortality. It's based on females. We have to assume it's going to be males. This brings in the climate change thing. So it's not like it's a steady change getting worse and worse. If you look at the 3 years there, you could look at 2006, 2008, and 2011. Those are outliers. They're very low pre-spawn mortalities. Those were cold, wet summers. So we're getting fewer and fewer cold, wet summers, but that really is linked. So it's not a direct cause. I'm going to get into a lot more detail about it, but it's a strong cofactor in pre-spawn mortality. We talked about dams, the challenge of dams. And here's a paper that we wrote a number of years ago. I'm a co-author on it. You can see my colleagues, Jim Peterson. This is a former postdoc of Jim and Carl's that's gone on. He left about 10 years ago to go at another university, modeling, et cetera, identifying optimal hauling densities for Chinook trap and haul. So I tell you, all these different systems, they have to truck these fish out. And what he found was that PSM was linked to specific transport events. The longer they were in the tanks tended to make higher pre-spawn mortality after they were released, but even more overriding. There was some, we don't know why, but certain days that there was really poor success on survival. So this is the kind of things that these guys do. Another Jim Peterson paper. These are field observations with a lot of math in them. So temperature, as I said, it's directly linked to pre-spawn mortality. But this is an interesting buffer the fish have. More fish in the river is the higher the pre-spawn mortality. Why is that? Is it transmission of disease? So in other words, if you have a poor year, you generally will have lower pre-spawn mortality. So if the stocks are low coming into the river, generally the pre-spawn mortality levels are going to be lower in those years. So that's a nice buffer that the fish have. So we can say, don't we need to worry about pre-spawn mortality? No, it's the numbers are, you saw from the Mackenzie River how bad it can be. This is an interesting phenomenon that's caused a lot of consternation and discussion amongst the fisheries biologists. More hatchery fish in the river equals more pre-spawn mortality, but not just because they're hatchery fish and hatchery fish aren't as good and all that, you've all heard that. It also reflects in the wild fish as well too in the river. We could talk about what could be a cause for that when we get time for discussion. What could be a link? So if you look into his papers, I'm not going to get into all the details on these, as they do ranking of models, etc. And you can see here that we see water temperature being high, Chinook mortality, they have their model, proportion of hatchery fish, this is highly ranked. So he has these rankings. What is influencing pre-spawn mortality? And if you guys are interested, I can give you all these papers. No problem. I can share. I'd be happy to share these papers with you. So why does hatchery fish equal more spawn fish? The last one I'm going to talk about with this, we're doing good on time, so I will be able to give my talk on our methods, and then we could take a break and have some discussion. It really does tie into what we're doing, and then we could start the second session with Corbin. I think this will work out well. Influence of stream temperature and human disturbance. So this is something, human disturbance, you think about, okay, the loggers are cutting the trees down around the rivers and all that, that's human disturbance, right? It's interesting here. A student, Catherine Carey, did her master's with us. Jim Peterson was her major advisor. So it's influence of temperature, more on temperature, and human disturbance on prespawn mortality in the Willamette Basin. And so again, these models, I'm not going to go through all this kind of stuff here. I'm just going to kind of give you the bottom line, is that activities so these fish make it up into these nice spawn, they pool up. If anyone's been out there, it's not just a little creek that goes all the way. There'll be pools, and they'll hang out in these pools waiting to spawn. When they made it up to these pools, and they got to still wait till we get rain to spawn, etc. That's a great place for recreating in August, for say. And they put up cameras, etc., and recording activities of people. This is just people floating around tubes, people jumping off rocks, people fishing, blah, 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 blah. And that unfortunately, there's a link to prespawn mortality by disturbing these very stressed fish in these pools at that time. That was not a popular result, if you can see, because there's a conflict of, people like to go. We always say, let's get out in nature and recreate, but leave the fish alone. That's kind of like the message that we had. And again, we talk about where we're really going to get into more is the prespawn mortality. So I'm going to move to my second talk. The timing is good. I'm not going too fast. I was worried about that I would be going too slow and not have enough time for discussion. We're going to have plenty of time for discussion. So now I'm going to end show and go to my other talk, which is right here. Okay. Now, this is something I've been lecturing for a long time. I'm transitioning out of lecturing a professor emeritus. I love my new position. I only have to teach 1 month a year, teaching a laboratory and veterinary I teach. One of my main courses is veterinary parasitology that I teach. And I don't have to go to committee meetings. Corbin's new at the university. We were driving up, they had a committee, the departmental meeting, and I could have zoomed in. I go, no, I'll get the lowdown from Corbin on the way up. I don't have to go and sit in a 2.5-hour meeting. I can just get the synthesis from Corbin. But I went through that too. So anyway, when I talk a lot with the students, so all these field observations, we're talking about, you notice I'm not saying causes, it's links, or associations. That's pretty much as far as we get with, often as far as we get with field observations. It's really important, and we're going to talk about my approach, our approach that we used. How do we bring it to more of a cause and effect? So we all learn in microbiology Koch's postulates. It's like something you learn in your first microbiology class. Here's Koch here, where you basically principles in determining cause and effect. So here, these aren't his postulates. This is what we do. Is field observations. They provide real world but complex interactions and many parameters. And then we do lab studies which provide pros and cons, have pros and cons about them. So that's our general principle, how we operate. Field studies, backed up with lab studies, in vivo lab studies. That's with live fish, or even the typical lab studies, molecular studies, etc. But we have great facilities at OSU for doing live fish studies, and that's really kind of what I view as where the rubber hits the road. And I still remember, Sydney will remember, when I was doing my postdoc with this guy named Ralph Elston up in Battelle in Squim, and I overheard him say in the hall, I kind of thought it might be an insult to me. He goes, oh yeah, Mike's an in vivo guy. That means I wanted, if you go through my papers, many of them entail doing studies with live animals in the laboratory, live fishes, to disentangle cofactors, et cetera, that we make in field observations. And so that's kind of the general principle. So here's our fellow Koch. I'm not going through all the details of it. Very simplistically, Koch's postulates were pathogen has to be prevalent in the animals that are sick or dying with a particular disease. You need to isolate the pathogen in culture. You grow it and put it back into fish, into your host, and cause the disease. That's a simplistic way, and there's even a paper that was written a number of years ago called Koch is Dead, in that things are much more complicated because they're multifactorial causes of disease. It took them a long time to culture the AIDS virus. They figured out that HIV was the cause of AIDS without doing Koch's, it's more associations and stuff. So what do you do when you have a pathogen that you can't culture? And we deal with those a lot because we deal with a lot of parasites that you can't culture. So it's more complicated, but it's a principle to start with to try to associate a pathogen with a particular disease. So assessing pathogen-associated mortality in wide populations. This is one of our fish that we looked at. I don't have the date on it. This is what we get, a pre-spawn mort. Hopefully they're going to be captured by ODF&W or students that are quite fresh. So that way we can do a lot more work at it. When they are old fish that have been sitting out in the sun, there's very limited. There's some things we can do, but there's a lot we cannot do with them. So we want to get fresh pre-spawn morts. So chronic pathogen-associated mortality, that's what we're working with our group is working with. It generally is parasites, but applies to chronic bacterial and viral infections, et cetera, as well, too. Often related to severity of infection more than prevalence. Just presence or absence, there's got to be lots of the pathogen there. Often chronic infections, seldom acute cause of death. And so when we start talking about these wild fish, a typical scenario, pre-spawn mortality, they're dead. They're big fish. They're going to be dead in the rivers. And then the otters and stuff will start picking on them afterwards. Many other fishes, you never see them because these underlying diseases causes reduced fitness. And the reduced fitness leads to, here's our Koch's postulates. We already talked about that. Leads to predation. It's a little bit out of order. I apologize. But these are in my talk. If you want to go through the details of Koch's postulates, I put that in there as well too. There's other people that have followed up. I've cited these a lot on determining cause and effect. These are epidemiologists. This guy, Sir Branford Hill. Here's a simple: strength of association, consistency, specificity. So this is making associations with disease, with cause of mortality without having to go through Koch's postulates. Biological gradient, coherence, plausibility, and that's really important as well too. So here's another one of his. This is a more detailed list. I really like this thing at the end. The whole thing should make biological and epidemiological sense. So you step back because you could do all this mathematical correlations. So like, for example, the situation where we talked about people recreating in pools with a fish. There is a biological sense. These fish are very stressed. They're getting ready to spawn. They're in fragile environments. They're jumping around in the pools and stuff. There is some sense to it. But if it's kind of like, okay, phase of the moon, or that's what people always joke about. Oh, it's the phase of the moon. And that's why I'm feeling, well, the phase of the moon may have a thing. But it's this underlying principle with all these details. It's taking a step back. And does it really make biological sense? So there's other papers that have been written by these guys. I cite them a lot. But let's get back to what I was talking about. I kind of got that a little out of order, and I apologize. So we have these fish. Our situation doesn't quite apply. They may be dying and then be killed by a predator, like otters. We don't have a lot of bears around and stuff, or seagulls picking on them and stuff, but they're on the way out. But if you look at smaller animals, smaller fishes, increased predation is a major cause of parasite or chronic pathogen-associated mortality. So they become sick, they're debilitated, they're slower. There's countless examples of, and I'm going to show you a graph that applies to this here shortly, you see on the upper left, you see this lion chasing the zebras. The general paradigm is the zebra that's most heavily parasitized, particularly if it's got, let's say, lungworms or heartworms or parasites that are debilitating its ability to escape prey are going to be the ones that are collected. And then I have a picture down here with a sea lion eating a salmon. The same principle applies with fish. So this is a paper that was written by a colleague of mine many years ago. It's a very simple paper, and it's cited a lot, and it was actually written as kind of for a symposium about how do we go about determining the impacts of a particular pathogen from field samples, beyond doing the lab work that I'll be talking about when we get to my other part, the end of this little section here. Necropsy, just going out and looking at the fish. I'm not going to go through all these. I'm going to go through a couple of these as examples that we use a lot. We use all of these in our program. Necropsy, just go out, the animals are dying. What's going on with them? What kind of pathogens? What's the state of the animals? Determining frequency of lethal infections. Okay, some of these we know are lethal. How prevalent are they? And then these other ones are a little bit more complicated, decreased prevalence of infection over time. That can be hard with a salmonid population. Same with decreased variation over time. Comparison of observed frequency of a combination of, so 3, 4, and 5 require very rigorous sample size and collecting them over time. This last one we use a lot is compare observed frequency to projected frequency of lightly infected animals. So it's kind of a fancy math, a program this guy Crofton did, but it works, and you only need one sample size. I'm going to show you an example of that. So just going out and doing necropsies, sampling sick animals in an aquatic environment, they quickly disappear. Hard to measure subclinical disease because you're only collecting dead animals. Many methods require sampling the same populations over time. And also, with the salmonids, access to some stocks is limited to endangered species. So often it's difficult to get anything other than just dead animals. So rarely find dead or morbid animals in the wild, quickly removed from the system by predators, I've talked about it, and does not provide the population level evaluation that we need. So an animal may have died from a disease. How does that reflect to the whole population? That's what really matters when we talk about, it's at the population levels that we're interested in. I'm going to go through a couple of examples here. Then we could take a break, answer some questions, and then we'll have Corbin start off the second thing. Here's determining frequency of lethal infections. This is down the Klamath. You may have heard about this parasite, Ceratodnova shasta, is a big program there. Dr. Bartholomew and Sasha Hallett and others at OSU have been working on this for a long time. And one of the things that they've done is going back and looking at the prevalence of infection in outmigrant Chinook in the Klamath River, because we know, here's some severely infected animals, we know if they have the infection, they're going to probably die within about a month. So you could predict mortality based on lethal infection. This is another area about patterns of disease and infection and chronic diseases and parasites. What we see with parasites is you don't see a bell-shaped curve. You see this negative binomial distribution. There's a bell-shaped curve on the left, the negative binomial on the right. Well, this tells us right here. So what this is telling us is that few animals have heavy infections, many with light infections. And it's those ones in the red that are probably going to drop out, where if it's a chronic disease that's making it susceptible to predation or not be able to migrate up the stream and everything. So it's that piece of the graph where we see it. And it's a negative binomial. And this is actually, they call it a law in parasitology. You look at any population of parasites, and it extends into the lab and sheep farms and everything. You're going to see, try to do this with my left hand, lots of animals with no infections or light infections. This is a healthy population. And then a handful with very heavy infections. And these are going to be the ones in a healthy population, a healthy wild population, are going to be picked off by predators. Yes? Oh, sorry. Okay. I'm trying to navigate this. Again, as I said, it's a law in parasitology. This guy Poland proposed this before. You see it in all populations of parasites. So you can use this, compare observed frequencies to projected frequency, and they call it truncation of the negative binomial, where basically you look at a population, you predict the level of infection, this would be numbers of parasites, based on lightly infected animals, and then the real world is this here, and this is this mark here where you run I'm simplifying it, you run a mathematical model, it'll give you the truncation point, and that's a point where you could expect to see parasite-associated mortality. And I'm saying parasites or chronic pathogen infections, mortality above that thing. So these are just some of the methods. When you hear some real data, not quite as clean as that, where we saw working down on the smolts on the West Fork of the Smith River, where we see a high level of the smolts from the main stem of the river had parasite burdens that would be predicted to not survive. I'm going to wrap this up with something that we do a lot. As I said, I was accused, I guess, maybe as a compliment or whatever, when I was a postdoc of being an in vivo guy. And we have great facilities at OSU, the Aquatic Animal Health Laboratory, the fish, another laboratory next door called Smith Farm, the Fish Production and Genetics Laboratory, where we have large tanks where we could bring salmonids in and do specific research. So this was a study we did back in 2011, correlating weight with parasite burden. And these are with our smolt study. Captive populations, they're easier to document acute mortality, easier to obtain fresh samples. You can control for all these confounding pathogens that we would see in wild salmon from the Willamette. And then you could do these defined transmission studies, fulfilling Koch's postulates. The con is you're removing all these confounding variables and you don't know which ones are really that important. Disease dynamics and pathogenesis in captive fish may not reflect the wild. So again, we had to show you that picture. I'm going to end here. Yeah, we'll have a few minutes. Here's a study we did with smolts. These are gill parasites called copepods. These are female copepods, and all these here are egg sacs. Right there, and those are filled with the next generation, Nauplii. So we see these, how are these associated with mortality? What's going on here? You collect the egg sacs, hatch out the Nauplii, expose fish in the lab, and generate data on mortality in juvenile fish, and seeing that we actually see a fair amount of mortality even before the parasites maturate. So it's giving us information to reflect on the wild. And now I'm going to end with this. So this is going to be part of the end of our second hour. We're going to be talking about where we really made some great inroads in understanding a gut disease that's very important in our Chinook salmon using laboratory transmission studies. There's Tamsin up in the top, Tamsin Pauley. She's the artist. Below her is Claire Couch, who really instigated getting this work going. An example of some of our outdoor tank facilities. This is at the Salmon Disease Lab, the ARL, they call it now, the Aquatic Animal Health Laboratory. And we'll talk more about that in the second hour. So I'm going to end with that. And we actually, as I said, we start talking about studies that we've done since 2009, right? All the way up to 2015. It's ongoing. There's a lot of new data. This paper just came out yesterday that on this work here. So this is hot off the press for you guys here. So it's trying to bring most up to date. We're not just going back to stuff that's 2 or 3 years old, and we have some other stuff that's come out on viruses and microbiome, et cetera, that is really quite new. It hasn't been published and really shared with anybody else yet. So you're the first to hear that. So I'm going to end with that. And you wanted to ask questions for a few minutes and take a short break? Is that how we're going to do that? Yes. Okay.
Good.
Hi, Cassia Quillinen. This is really fascinating. I think it's way over my head, so I have a more down-to-earth kind of question.
Sure.
You have all these fish that have various viruses and pathogens and so forth in them that you're studying. Presumably, some of these fish get caught and eaten by people.
Yes.
What's the effect on people, do we know?
Yes, we do. So there are a handful of pathogens that are transmissible to people if you eat the fish raw or undercooked or cold smoked. One of them is an anisakid worm, and there's a few tapeworms, etc. So as far as human zoonotic diseases, human health diseases, we only have associated a few parasites and these parasites generally would go through a marine mammal or a bear or a bird or something like that, where they're designed to be able to live at a warmer body temperatures. The bacterial and viruses are I'm not aware of any that will infect people because our body temperatures are just way too warm for them.
Good morning. My name is Eric, and the question I had for you,
Hold the mic to your face, please. Thank you.
with global warming, I know that many of the salmon do not fare well in warmer water temperatures, especially the coho. And do you have any projections as time goes along and the global warming increases or continues, how that's going to affect the salmon stocks?
Well, I think that's a very broad brush answer is there's a lot of concern about how that's going to be affecting it. And I think it's going to be each species. Like you say, I don't know, I've done very little with coho down here when I lived up in Canada before we did work with coho. So it's going to depend a lot on the impacts of global. It's not going to be evenly distributed. It's going to be, with climate change and global warming, it's going to be system by system and species by system. Generally, I would say in general, it's not going to be good, but it's going to be bad or horrible, depending on or an impact, and some will not be. It's interesting. There may be some systems that are just not affected. It's going to depend a lot on snow melt, and as we know, climate change. We're worried about having less snow accumulation because it's going to be the temperature. Like I said, those 3 years. They weren't the first 3 years. There was the 2008, 6, and 11 or whatever. But they must have been very high snow melt. So there's all those different, and so that's going to affect different systems. So it's a general trend, but it's going to be much more granular. At each different system is going to be impacted. At each species is going to be impacted on its own.
Thank you. My name is Brad. Confession first, my personal background is in global health, infectious disease epidemiology. I was a little surprised to not see any regression analyses. Is that because these data are too disparate and too small sample to come up with R squared, or was it that for this audience really R squared?
Well, if you looked at our laboratory study, we did have regression analysis on that. Oh. The field stuff, it becomes more challenging, but certainly we have done that a lot. Jim Peterson is the modeler and stuff, and it's often difficult to do that, particularly because these things are it's a negative binomial distribution, so it's another challenge that you have to do these non-parametric tests and things like that. Mm-hmm. But no, I'm not saying we have a lot of data where we do regression analysis on other on some of these endpoints. Like I totally agree with you, like pathogen burden and versus the other pathogen burden, let's say, versus some other fitness endpoint or something like that. So
is there a strong R squared?
Moderately strong for field work. I mean, as epidemiologists, we can go back. Let's look at. So here's
maybe a word about what R squared is.
So here's R squared. We were pretty happy. So here's exactly what you're talking about: pathogen burden on weight, and we've got an R square of brood year 2007 0.22, brood 2008 0.34. For this kind of data, we were pretty pleased with it because this is our endpoint is size. Lots of things are going to affect the size of the fish, and so having this correlation with parasite burden, we were pretty pleased with that. You show that to someone working in the lab, with defined exposures on it, they go, "Oh, that's a horrible R square." But this is actually so R square. The higher your R square is, the more correlated your two findings are. So an R square, nobody ever gets 1, but an R square of 0.8 is fantastic. So with your field epidemiology, you're doing field work as well too. So what kind of R squares, if you see an R square of 0.2, 0.34, do you discard that or? Okay, thank you. Thank you.
Just a general question. My name is Janet about federal funding, what you have any staff cuts so far and what you think's going on?
Well, this is very timely because Corbin, Dr. Schuster he's the lead now. I'm stepping back. I'm on the grant, but he's just been awarded a Sea Grant proposal that's funded. That's Oregon Sea Grant. As far as we know, it's still funded, right? And it was I don't care. I think, what are you going to do, fire me? I'll be honest. It was a dance that we have to do, particularly because we have Native American stakeholders are involved with this project. And at the state level, they loved it. They're worried about, at the federal level, not liking it. But we have stakeholders that go beyond we have what's the sports fishing group, Corbin? Yeah, the Coastal Conservation Association, which is sports fishermen. They're on board for collecting fish. And of course, one of the biggest stakeholders are the state agencies, Oregon Department of Fish and Wildlife. So it's a lot of wordsmithing that we needed to do to navigate the environment right now, but things are still looking, they haven't shut down Sea Grant yet. Yeah, and Sea Grant, so it's challenging times, but we're not jumping off buildings yet. As a research scientist, you have to be optimistic because so many experiments fail. I'm showing you all the stuff that works, right? There's lots of things that do not work. Well, all the fish, I had a technician, she was great. I'm not gonna say her name because maybe some of you know it. She was like a Debbie Downer. We run these experiments like this, like running them for 6 months. And I remember one time I said, okay, monthly samples. Our last sample is on Monday. Everything is looking fantastic. This is what a success on this experiment. She goes, well, all the fish could die this weekend. And I bring that up because that is the truth. Corbin knows that from his work with zebrafish. Well, he pulled off a fantastic experiment, and it was his first big long-term experiment. And I don't think you knew how fortuitous. I'm not going to say lucky because you did a good job on it. Running long-term experiments in the lab with diseases that are difficult to transmit, it's challenging. So I kind of got off track. So you have to be optimistic if you're going to be a research scientist.
My name is Neil, and I'm just curious, you didn't talk at all about pollutants in the water.
Okay.
I'm just simplistically, have the issue of pollutants in the Pacific Northwest gotten better or worse over the last 20 years?
That's not my area. That's another area that I guess we should put in there. I'm sorry I didn't put that on that list. That's not an area that we've been working with. We have not related pollutants per se, but there hasn't been that extensive investigations on persistent chemicals in them. There's a particular phenomenon that's occurring up in Washington with coho salmon, with urban runoff from tires, etc. And they've shown that that's devastating to this one particular run of coho that goes through Seattle. So that's a very unique situation, and it's pretty convincing what they've shown about how damaging that is. But as far as our situation in the Willamette, and I was involved with another project on looking at skeletal deformities. It's hard because we don't really have a point source pollutant that you can nail down. I'm not saying it's not existent, but it's much more broad. Like you talk about agricultural chemicals. It's outside of my area, so I can't say it, but that's something that should be put on the list too. Sorry.
Thank you. We'll take a 10-minute break now and then start with the next session.
So we're going to get rolling now with Dr. Corbin Schuster. It's my pleasure to introduce Corbin. He came to my laboratory, what year, Corbin? 2018. And did his PhD in my laboratory. Yep. And then went on and did a postdoc at University of Oregon working on zebrafish, diseases in zebrafish. That's what he did with me. And then went on took a position as a faculty member at Heritage University because he can talk a little bit more about his background. He's from the Yakima Nation. And then we stole him away from Heritage this year. Brought him back to Oregon State University in our Department of Microbiology. And as you can see, he is hoping to continue his work on zebrafish, but is already getting heavily involved in the salmon work as well too. So I'll turn it over to Corbin.
All right. Thanks, Mike. Actually, kind of a funny story. I'm originally from Washington. So, and both my parents are Washington State University alums. So when I came to Oregon State, I was instantly called a traitor, but it's fine. Dr. Kent actually was what attracted me to come to Oregon State University. And he asked me today to talk about the community-centered research approach that we're taking with our newly funded Sea Grant. So we were awarded about $250,000 to continue this pre-spawn mortality issue. But we have started to engage, or take the approach to engage those that are on the ground. So working with our tribal fishers, working with our community recreational subsistence, non-tribal fisher groups, as well as our state and federal agencies. So both the United States Fish and Wildlife Service, as well as Oregon Department of Fish and Wildlife. And so today I'm going to talk a little bit about what that approach sort of looks like. And I'm going to use our tribal partnership sort of as a case study, but also this has turned out to be true for each one of the groups that we have collaborated with. Just to give some positionality and connection to the area, I grew up, as I'm an enrolled member of the Yakama Nation, descendant of the Warm Springs. I grew up fishing the Columbia River and its tributaries. So I grew up running around Hood River, the Dalles area. I also, Willamette Falls, a beautiful picture of Willamette Falls, grew up harvesting lamprey, Pacific lamprey, as well as Chinook salmon at that traditional site. So I grew up sort of from the perspective of being engaged and concerned with how the fishery has been sort of developing over the past century. And even just within my lifetime, we have witnessed the extreme declines and the sheer number of fish that are returning back to our upper tributaries in the Columbia River. I'm sorry if this didn't come out too well, but really the question as to why community engagement in salmon research really matters. In the Willamette Basin, we have had in some years a 90% pre-spawn mortality. So you had a significant amount of that returning stock completely die off, leaving about a 10% pool of fish for that single year. So it was an extreme bottleneck. In the Columbia River, we've seen an upwards of 65% in some years. So we've had some extreme pre-spawn mortality events, and this really reaches not only to the tribal community, but to every community in which that sort of economically and lives off of the Chinook salmon. In regards to the sort of history, I'm not going to give a history lesson here, but in regards to the history, it's usually lost treaty rights as well as food sovereignty. The culture of tribal peoples in the area are deeply connected to the salmon, and in fact, even our languages are connected to the salmon. So you talk to some of my relatives up in the Spokane area, upper reaches of the Columbia River, and they've talked about how they have lost some portions of their language because they no longer have Chinook salmon in those upper regions, in those upper tributaries in which they were ancestrally stocked with. And so with that, it's erasure of these cultural practices, these ceremonies that these folks engage in, and some of those traditional practices. We talked to some of the tribal elders with the Warm Springs as well as with the Yakama Nation, and they talk about some of those traditional ecological knowledge. I know it's a sort of buzzword, that TEK, but the idea of that traditional management strategy before hydroelectric dam development and before colonization. And some of those practices included pulling nets, pulling fishers off of the river in order to let those genetic lines that are going to those upper reaches of the Columbia River, to let them pass and let them be able to spawn in those upper reaches. Of course, I think we can all relate to the economic impacts on fishing communities, whether we're in Newport, whether we're in the Dalles, Hood River, Cascade Locks, any of these areas, in regards to just how tied to the river and to not only the Columbia River, but as well as the Willamette Basin, these communities are. And so with that, you also get Chinook salmon that represent a sort of keystone species and an ecological indicator species. So with the lack of the salmon in our systems, it indicates that our ecosystem is no longer, at least at the moment, isn't sustaining the populations at which it used to historically. So our approach here is to engage all of our community, to engage all of our stakeholders, where we can merge Western science and generational knowledge, both traditionally as well as over time, as these folks, as these fishers have gotten to know these river systems through several generations. And I come from a very molecular side. I work at PCR, sort of like the COVID assays, right? So we developed some of these molecular assays to be able to detect and understand transmission dynamics and prevalence across systems. And so really the question might be like, well, how are you going to engage community in such specific work that requires such high expertise? And so it was a really cool approach that we took in regards to starting to understand what it means to engage with these communities. And I spent the past 3 years meeting with these folks and talking about what their concerns were. What were the issues that they thought were important that we approach as researchers? So instead of moving from an extractive, this is what I want to do, this is what I'm going to do, and not really give mind to what the community input is. Instead, we were asking what that community engagement might look like for these communities. What would meaningful science be to the Yakima Nation, or to Warm Springs, or to the fishing community in Newport? What does that look like? And everyone sort of had a different perspective. And I know in this sort of political climate, or landscape, it might seem odd to include all of these different perspectives, but it's really, really interesting because you get different concern. You get different concern from a tribal fisherman's background versus a commercial fisherman's background, right? But the collective concern is that we're concerned about the decline that we're seeing in our systems. And so we wanted to sort of build on the idea. Not only did we want to engage with the community and provide some meaningful science, but we wanted to start to include these communities in the decision making. What does the data mean? How do we interpret it? And how can it be useful to these communities? We started to want to, but our main goal really was to build long-term sustainable relationships. So not just come in for the duration of our grant period and collect data and sort of leave these communities out in the cold, but rather to create a surveillance network. And Dr. Kent will talk about some of the more specific science here in just a minute. But our idea is to start to characterize the distribution of both what we're seeing with these pathogen-associated prespawn mortalities, such as enterocytosome Schreckii, as well as the viral inclusions that have yet to be characterized. But a lot of our Sea Grant work is really to work to characterize the distribution of these parasites and these pathogens within our system, and what that's really looking like in regards to long-term management of the fishery. And so part of that component of engaging with the community is understanding the reciprocity and the trust-building capacity with these communities. And so we started, as I mentioned, with identification of the community concerns before we even started our research, and actually before we even submitted the Sea Grant proposal. So we really wanted to be able to identify what were the major concerns that folks were willing to engage with us on. And so our goal is to be transparent, not only with what we want to do, but our methods as well as our limitations. But another really important component was to show that we have a sincere investment in our community. And with that, we want to engage with the tribal fishers, we want to engage with our commercial fishers, our recreational subsistence fishers, by actually compensating them for contributing samples to our protocol. And so part of our grant is to provide that compensation. We would provide training, we would provide all of the necessary equipment that they would need to be able to provide samples. And it's a really unique perspective with our fishers specifically, because they're able to get into reaches that frankly, myself or Mike or anyone in my laboratory might be limited to actually go out and collect those samples from, right? These people are all on the ground. They know where the fish are gathering. They know where the fish, typically are. And so it's it's a unique perspective that we wouldn't get otherwise. And it's another thing to also engage with students, to engage with the next generation of researchers and scientists from those communities, and to include those both in my laboratory, so training students. Bailey Morgan, she's from Portland, Oregon, but she has grew up fishing the area, and she's very engaged with our fishing communities in the area. So we had a sort of strategic partnership structure, is really multi-stakeholder. Of course, with OSU, our CCA is the Coastal Conservation Association that was alluded to earlier. These are sports fishers, recreational fishers, as well as subsistence fishermen. We have our Columbia River Intertribal Fish Commission, which is going to serve the Yakima Nation, Warm Springs, Umatilla, as well as the Nez Perce. These, as well as the Northwest Indian Fish Commission. The Northwest Indian Fish Commission serves the upper tribes up in the Puget Sound area, Seattle area. But we're starting to get some understanding and characterization of the spread of Enterocytozoon schreckii, which is a parasite that is associated with adult salmon enteritis that Mike will talk about here in just a sec. But we're not seeing it in those upper regions, up in the Puget Sound area. We're not seeing it in the White River area, so around Muckleshoot or Auburn. These areas are also, like these hatcheries are glacier fed, so they're very rich riparian zones, but it's really interesting dynamic. And so this year we started to extend more along the Columbia River Basin, including Carson. And some of that data is a little bit preliminary, so I'll be a little bit conservative with reporting on what those results look like. But it's a really interesting dynamic because this year might be the first year that we've actually identified some of the parasites in the Washington stocks. So we include the tribal nations, the state and federal agencies, academic institutions, OSU, where we're providing laboratory diagnostics, molecular tools and analysis, as well as student training. And then there's also our community organizations and our community partners. And our goal here is to meet with these groups, is to hold consistent biannual workshops where every one of these groups will be invited and asked to interpret some of our data and to talk about what this means on a management level. And so these two gentlemen are both of my past researchers. Both of them come from a tribal fisher background. Christian Serrano was in my lab this past summer. He was helping me develop some molecular assays for Enterocytozoon schreckii. It's one of our parasites. And then Lance Martin, he helped me collect some of the samples from the Washington hatcheries. But really, these students, they sort of understand the cultural and place-based nuances that really help to build trust. And so these students have come from the community and have helped me as a professional connect to their respective communities. And so they really help to bridge the gap between academic institutions and in these populations. And so that's really the summary of what I wanted to talk about today, was sort of that relationship building capacity for this approach, this unique approach to the Sea Grant and to our pathogen discovery, as well as the field coordination that it requires to be able to work with our tribal fishers, as well as that knowledge exchange between the academic institutions and the communities in which we serve. And I will end there with this slide. But really, our overall goal is research that serves the salmon, serves the people, and honors the relationships that have sustained both for hundreds of years. And if that, I'll pass it over to Dr. Kent.
That's funny. Okay, so what I'll do now is I'm going to pull up my talk and talk for half an hour, 20 minutes, something like that, to really kind of get into. You've kind of been going around this thing about what a core area where we're moving forward with our research is on this disease that we call adult salmon enteritis, that we find as an underlying consistent association with prespawn mortality. I'm going to end this here. Okay. So now for this part, I'm going to talk about we're microbiologists, parasitologists, pathologists, getting more into the specifics of pathogens that we see, and then finishing up with the specific. You've heard about this one pathogen, enterocytozoan schreckii, and some of our most recent findings. So here's our team again, a long list of researchers, microbiologists, fisheries biologists, microbiome experts. We've got some interesting information on the bacterial microbiome. That's the bacterial community in the intestine of the fish. Ecologists, fisheries biologists, and you see that name, Carl Schreck. He is I would say the grandfather of stress physiology in salmonids. World-renowned scientist, and that's why we named this parasite, and you'll see soon, enterocytozoan schreckii. We named it after him. That's one thing nice. We describe a lot of parasites. He thought we were kidding. He goes, I always thought I'd have an animal named after me like an eagle or a salmon, and I got this annoying parasite named after me. Again, you saw all these supporters that we've had. And so we have our list of I'm picking up where I left off, all these different things, and we should include pollution on here as well, too. We have not done a lot of work on that. Other areas where they have linked anthropogenic, man-made pollution to prespawn mortality. And then we go to disease, and that's what we're interested in mostly. I mean, amplification of endemic pathogens. How about novel pathogens? And diseases and lesions that are not associated with pathogens. So our approach includes histopathology, which allows you to look at things that go beyond just looking at the pathogens. The list, as I said, these fish are immune compromised, so the list is quite long of the different pathogens that we find in these fish. I'm not going to need to go through explain all of them. I'll show you a couple of pictures. Gill parasites, the Microsporidium enterocytozoan schreckii. That's one that we're really spending a lot of time on. It's a very interesting organism. Bacterial kidney disease, Aeromonas salmonicida, a long list of pathogens and diseases. So this is some examples of pathogens and pathological changes. This is what we do. Is we go in the field. Here's that picture of a salmon with their gills all covered with these copepods. Here's a fish at a hatchery with a huge spleen and a large kidney. We're going to be taking samples of that for pathology. Down below is a wet mount of taking the kidney and just squashing it on a microscope slide and counting those spheres, which are the developmental stage of a parasitic trematode that causes salmon poisoning in dogs, and that's a whole other story as well too. So if you ever heard about salmon poisoning, this is a worm that carries the bacterium that causes salmon poisoning. So we do our work is led by histopathology. And why is that? Because we don't have an a priori list of diagnostics. We have a list of pathogens. What happens if there's something new? Also, you can see this liver damage is consistent with a disease we described up in Canada called net pen liver disease that's caused by a toxicant. So that's going to get those. Here's a tissue reaction to a parasite called parvicapsula. So this is going to give you the assessment of the severity. And here's a novel parasite that's not on our pathogen list. We call it a rosette-like protist. There's an organism called the rosette agent, but it's never been described in Chinook salmon from the Willamette. We see by histology a parasite that's consistent with that. If we just had one of Corbin's PCR tests, you can't make the PCR test until you know what the parasite is. So we lead with histopathology, an expert molecular biologist like Corbin carry it on to making PCR tests, etc. Provides information on associated lesions, includes non-pathogens, as I said. And then we save stuff back, frozen for culture, molecular test, whatever. This is a complicated slide. This is just showing you over the years an example of our collections. Now we're over 1,000 fish that represent healthy fish, fish at spawning from hatcheries, pre-spawn mortality fish, and post-spawn fish. And so this only goes up to 2021. We're continuing this work, so we still have a few more years of data. So that's why we can add to that list and say we have over 1,000 fish. Here's a number of the locations that we've looked at. As you can see, most of them, Willamette Basin, various Mackenzie, various contributors, Round Butte, and down at the bottom, Carson Hatchery are off the Columbia. So they're not off the Willamette system. Sandy River comes off the Columbia. And we got one sample of fall Chinook. We're concentrating on spring Chinook. We opportunistically were able to get fall Chinook. And then those two places that Corbin mentioned up off the Puget Sound area would be White River Hatchery and Minter Creek Hatchery. They're not fed off the Columbia. They're fed off Puget Sound. So here's our list of all these pathogens. And then remember I said we can do histopathology. We found a lesion that was a pathological change that was so common that we decided that we need to look into this further. Is there a link of this to prespawn mortality? So this is what we're talking about. Here's the intestine of a salmon, sort of like ours. Here's a stomach, and here's the lower intestine. That would be like our large intestine. The small intestine, instead of being a long, like 25-foot long tube, which is a small intestine, it's a bunch of blind sacs called ceca, as you see here. So these accomplish the same thing as a small intestine would do, but there are a number of blind sacs. So that's the basics of the digestive system. We start looking at the histopathology on these fish. And I remember showing a slide like this to a fisheries group, and a colleague said, don't show them histopathology. They're not going to understand this. But I think you could see a dramatic difference between the normal and a PSM fish, how it's lost. The villar structure, your normal gut has fingers on it. It's lined by an epithelium, which is a layer of cells. We call them enterocytes, because enteric. And that's their epithelium. Minimal inflammation down the next layer. And here's a prespawn mortality fish. The epithelium has been changed dramatically. We call it dysplastic, or completely lost here. So very different scenario in these fish. A little higher magnification. I'm not going to get into this, a lot of details for this audience, but we describe the lesions very carefully. And this is midsummer fish, and most post-spawn fish show this. All the pre-spawn mortality fish show this. So basically, I'm going to get to a graph here. We follow this here. Let me go back a little bit. Talk about our locations where we've seen this gut change. So now these are the areas where we have not seen it. Let's look at where we have not seen it. Those two hatcheries off the Puget Sound, and then the fall Chinook from Sacramento. So all the fish that we've looked at, all the runs that we've looked at that are linked to the Columbia or the Willamette Basin, we have seen this disease in one degree or another. So one would say it's so common, it's just a normal phenomenon of natural senescence. These fish are destined to die anyway. So this is a normal phenomenon. But if you look at White River and Minter Creek, we've looked at quite a few fish from there, multiple years, their intestines look completely normal, like the picture that was on the left there. So it's not just a normal phenomenon of spring Chinook. So again, this is showing a general idea of the different areas where we see it. I just explained that. Columbia River and Willamette River, the limited up at Puget Sound. There's a lot more places we need to look at, but that's a start. So we moved on looking at these lesions, and here's this parasite, Enterocytozoan schreckii, and another parasite that's very common, Strattonova shasta. Those are the two gut parasites that are really common. So we had to try to disentangle the role of those parasites in this disease. And then also enteric viruses. I'm going to kind of get to that. We're just kind of scratching the surface on the viral work at this time. Here's Strattonova shasta. It's a parasite that requires a polychaete as an intermediate host or alternate host. It cycles through this. And this is the parasite up in the epithelium here. It's quite common and down in the lamina propria. So this certainly can cause lesions. And here's our new parasite, Enterocytozoan schreckii. We just described it. We've never seen it in fish other than those fish that have the enteritis. We call it adult salmon enteritis, ASE. So if I say ASE, I'm referring to the gut disease. And and as a parasitologist, and Corbin did his PhD on Microsporidia, so he knows this very well. These guys develop in the surface cells, the enterocytes. These are presporogonic stages. They ultimately form spores, and then they're sloughed off into the lumen. So what's really interesting about this parasite, so here's a paper we described, Enterocytozoan schreckii, a fish parasite. The closest relative to this is Enterocytozoan bianusi, And remarkably, this was first described from AIDS patients, from humans with AIDS, back in 1986. So where is the link? Humans are profoundly immune compromised, and so are adult Chinook salmon. And they're not only morphologically very similar, they're molecularly similar, and they cause the same kind of infection. So here's salmon on the right, on the left. Here's some sloughing enterocytes of a human with Enterocytozoa bianusi. So quite an interesting connection between the two of them. Also along the way, when we were doing electron microscopy on the Enterocytozoan, looking to describe the morphology for our species description here, getting all these development, you don't need to go through all this, but this is electron microscopy of the spores, etc. Lo and behold, we found a virus in these fish as well too. So this complicates it. The world is complicated. And now here's our get back to regression. Here you get to see another regression curve, right? So this is even more complicated than those other regression curves. So if you take out the pre-spawn mortality fish, we see this in the Willamette system. We see this over time. We see this the severity of the disease scored by the amount of the epithelium increasing. So this is a negative regression. The longer we go, the less epithelium they have. And here, these are a whole bunch of post-spawn and fish from hatchery. We have lots of fish from hatchery. Some of them look pretty good, but lots and lots. It really gets busy down here. If you look at the pre-spawn fish, they start having very little epithelium left, but much earlier in summer. So basically what a pre-spawn mortality fish looks like is a post-spawn fish. So everything's happening too fast in the Willamette Basin. Then we lay over on top of it that this disease I'm describing, which is so common, doesn't occur everywhere. Here's another way of looking at that regression. Here's the pre-spawn mortality fish that we collected earlier in the year, and here's a regression line showing the normal loss in the fish that make it to spawn. A couple of other examples. Way of looking at the data here, the pre-spawn mortality fish. Well, here's some fish that look very normal. I would contend that these fish actually came from fish that were holding in tanks for some tank studies with other bacterial problems. These are interesting here, the live fish. Nice normal guts up here. Outliers, these guys down here, they're captured live, let's say in July. I would contend that these may be fish that would be dying in a few weeks later or a month later, would not make it to spawning. That's our hypothesis. How about temperature? We were all concerned about temperature. Actually, we did some studies, and this was published. Benda is first author, Carl Schreck, myself, and others. Holding them in tanks. Here's our live studies again. These are just captured big fish from the field, holding them in cold water, and then versus fish from the river and the hatchery. What we found is basically the same amount of loss of epithelium. Doesn't matter if you put them in cold water or not. So the rate of losing the epithelium is not, from this study, was not pushed by cold water. However, the fish that were held in cold water with the gut, they had the same amount of gut disease, they were 7.9 times less, or the fish from the river or the hatcheries are 7.9 times more chance of dying than fish held cold. So basically, what this is telling us is you could take these fish with these severe gut diseases, put them in cold water, and they'll still make it to spawn. But you'd have to cool down the whole river, right? This is running through the whole summer at 12 to 14 degrees. So ASE was no less, but cold water reduces mortality. Okay, so we're going to wrap up with what I like to do. I'm weaving back and forth, being the in vivo guy that I am, transmission studies. So this is back. I showed you this slide earlier with Tamsin, Dr. Pauly, and Dr. Couch, and using some of our outdoor tanks in the facility. So when we see this phenomenon of the gut disease and we see it not occurring in some place, it makes us hypothesize that this isn't just something that happens to all spring Chinook. So could it be caused by an infectious agent? We already saw this Enterocytozoan schreckii that's only found in those fish, but we still haven't fulfilled Koch's postulates by any means. It may be just along for the ride, just like Enterocytozoan bianusi is with AIDS. AIDS is caused by a virus, but the humans are immune compromised and therefore much more susceptible to enterocytozoan B and UC. So this paper just came out yesterday. So getting timing on that, it was just published online yesterday on our transmission work. I'm going to go through it fairly quickly, and you could ask me questions after. So what we did is we took small fish. Remember I told you they're severely immune compromised and they don't eat. So we had to mimic with small fish what would be going on with adult fish. So we fasted them. We had, summarizing a couple of experiment stuff, but distilling it down to the broad brush here. Fasting them, we had fish that were fed none. We had controlled diet at 3.5%. And then we immune compromised them with implants of dexamethasone. It's one of these steroids that some people are treated with when a physician wants someone to have a steroid, they may prescribe dexamethasone. And it works very well in fish. What we do is we mix it up with Crisco and then inject it into the abdominal cavity of the fish, and it solidifies. Then that allows for a slow release of the dexamethasone over the length of the experiment. That's what's going on in the wild fish. Not just giving them one quick shot or giving them some pills or whatever. It maintains this high levels of immune compromise. So we did that. Where are we going here? And then here's our timeline. We added ASE exposures. Basically, we got guts from fish from the Willamette hatchery that had the disease, ground them up and intubated the fish. This is Tamsin's nice artwork. This looks kind of like hamburger, right? These are ground up of salmon guts here. Run through a screen, intubate the fish, and then follow the disease. Oral gavage. And this is what we follow. So mortality, this is telling, what we've learned this and from other experiments as well too, is that fasting really doesn't hurt the fish. As far as killing the fish. They're not putting on weight or anything. So these are fasted fish. These are fish that are given the steroids. The ASE, the steroid, the dexamethasone without the agent, without fish guts. Here's with fish guts, and they didn't even make it to the end of the study. So, and this was statistically significant, this decline in survival. So what did we see? This was really the most exciting part, is when we start looking at the recipient fish, is that we were able to recreate gut lesions that were, here's normal, gut lesions that were consistent with what we would see with ASE disease. Dysplasia score, enteritis scores, etc. So we just scoring these. And basically, what I want to get to is where the rubber hits the road. We start looking at histology, and we found the recipient fish, we grind up the guts, and they developed enterocytozoan infections. The life cycle of most microsporidia are just direct. You just eat a spore and you get infected. Remember I showed you the Serrata nova shasta has that complicated life cycle that involves this polychaete worm? These fish all had Serrata nova shasta as well too, the donor tissue, the stuff that we ground up, no transmission. So no Serrata nova shasta in these recipient fish that developed the disease. So it's pretty good evidence that Serrata nova shasta, which is extremely common in many of these fish, is not the cause of this. We haven't ruled in or out enterocytozoan, but for certainly enterocytozoan was transmitted. Then here's another twist. Remember I showed you early on, I said, we found some viruses when we looked at the fish. I'm going to lead you through this to say, only a histopathologist would really appreciate this, is viruses that infect the nucleus will cause inclusions. And looking at the histology, these things here are very consistent with a viral inclusion. If you look at cytomegalovirus in an AIDS patient, they're going to have inclusions that look similar to that. So you can't say for sure it's a virus, right? But it's suggestive of a virus. So then what we did is I'm getting near the end of this, so we're doing good on time. So colleagues of mine, they're listed on my collaborators, Dr. Lou Weiss and Peter Takvorkian at Albert Einstein College of Medicine. Lou is a longtime friend. He works on microsporidia and AIDS patients, et cetera. He's an MD at the medical school there. They run this program. It's done at other universities, but he's my buddy, so he ran it for free for me. It would normally be very expensive. Correlative light and electron microscopy. So basically, we're giving him slides with these inclusions, and then they look at them under the electron microscope. It's not the greatest quality because it's not processed the way you'd want. And then inside of those inclusions, we're finding viruses. By the way, they did the clam. They go, oh, there's your microsporidium. We would identify a microsporidium by these tubes here. Anyway, so along the way, we showed the enterocytosome by light microscopy as well, too. Cell culture, this is last year's. We're repeating this again. This shows the use of the help of collaborators. This is Melissa White, Oregon Department of Fish and Wildlife. They're housed in Nash Hall. And they're not Oregon State University, but they've a long presence on campus. She does cell culture. Here's a cell line right here, a monolayer of fish cells called EPCs. And this is normal. She puts our material on there and she goes, oh, I'm seeing CPE, cytopathic effect, balled up cells and stuff. And she was able to keep passing it. So she said, well, it looks like a virus. We did electron microscopy on the material and found a virus. And then last, this is some of the most recent work, is our colleague Tom Walsick up at Washington State. He's a professor, a fish virologist. He's finding virus sequences in our material. So a lot of stuff on the viruses, no specific answer yet. Then I'm going to wrap things up with, what about bacterial agents? Here's some bacteria in one of these fish with a disease. See where the B is right there? So from the bacterial approach, we think about the intestines of a normal fish or human is going to be loaded with bacteria because we have fecal material in there. It's helping digest the material, break it down, etc. These fish are not feeding, so there's not going to be a tremendous amount of bacteria, but there are still bacteria there. Our colleagues working on the microbiome, it's very complicated, but basically they're looking at patterns in bacterial communities in affected fish versus unaffected fish. And this is, again, right off, Corbin was in the conference call with us. This is just like 2 or 3 day old data. They are showing remarkable segregation by normal fish. That's Minner Creek, White River, being different than these brown ones. These are the fish with the disease. So again, another lead. We're not saying cause. This is certainly not cause and effect. This is what we call dysbiosis, a change in the microbiome. We haven't identified a particular bacterium that may be contributing to the lesions, but when you have a sick gut, your bacterial community is going to change, and that's what we're showing here. So I think what I'll do is I'll summarize here, and then we can have some. They're perfect on time. PSM is linked. Prespawn mortality is linked to several abiotic and biotic causes. It is linked to this adult salmon enteritis in the Willamette area, not up in Puget Sound. Willamette and Columbia River. We still have a lot more places to look at, but we have pretty extensively looked at the Willamette River, and we can see a pretty strong link. We've shown from the lab studies that ASE-like lesions are caused by a transmissible agent, and what do we see in the recipient fish is enterocytozoan schreckii and novel viruses. And we also just recently see ASE is linked to specific. You see, I keep on saying linked. I don't say caused. Linked to specific, to distinctive microbiome bacterial changes in the gut. And it has a limited geographic distribution. We have not seen it in rivers that come off Puget Sound. So not simply a background phenomenon of Chinook salmon across the Pacific Northwest. We've looked at some fish in BC, very limited samples, and we haven't seen it in those as well too. So with that, I'll end our talk 2, and I guess leaving us some time for questions. Is that correct? I should say talk 4. Corbin, why don't you join me up here? And you can ask Corbin questions too.
My name is David. So tell me again, when did you first start seeing the E. schreckii?
When we went back retrospectively, that's one thing nice about pathology. They're back in blocks and slides. We went back and found it in our early, we started working on this in 2009, and that's when we found it. And, you know, people have been studying fish diseases in the Pacific Northwest for a long time, and Chinook salmon, not as much on what we're doing, looking at adult salmon. Lots and lots of work on fish from hatcheries. When I was up in Canada, and people are still doing a lot of Chinook and marine net pens, no one has ever reported this parasite until 2009. I'm not saying it recently introduced. I wouldn't be surprised if we could find some archive material from the Willamette system that goes way back. We probably would find it. But it's still, so far, only linked with this gut disease with salmon.
Hi, my name is Anne, and I'm a French teacher, so, you know, that's the background. I'm just getting the nuances, you know, here and there. So it's colder in Washington. That's where my thinking goes.
Oh, cold.
It's colder. The average temperatures have to be a little bit colder in Washington.
Well, no, you know, talking with my colleagues from the White River, it's plenty warm enough there. Minter Creek is an interesting phenomenon. We can actually get the temperature. We can get those because it's a short river and they put them in tanks and hold them over summer. But we can see the studies that we did holding fish in cold tanks on campus, they still develop the disease just fine. So it's not, the two things that make the two negative hatcheries stand out, they're not associated with the Columbia River or the Willamette system, and they're on short rivers. That's one thing that's rather interesting. As Corbin said, they're glacier-fed, relatively short rivers, whereas, you know, like fish at Carson and Deschutes, Round Butte, all these other places have much, much longer journey to go. They're in the water just as much time, because they confirm these are not fall Chinook at White River. These are spring Chinook. So they're coming back and they sit at the hatchery on this glacier. You remember we were there, you could see the mountain right there. They're on glacier-fed water through the whole summer. So it's not just cold, but it's still plenty warm enough too. So it's complicated.
But the ones that are in the cold water don't die.
That's right.
They have it, but they don't die.
Exactly.
So, any other questions here?
All right, Cassia again. To follow up on what you just said and what Anne's question, I think, was leading to, the fish that are still out in the ocean, you catch them too, I assume. And do you still find the same prevalence of these bacteria and viruses and pathogens? Or has that comparison been made yet? And so is it mostly once they get into the river system?
Most of the pathogens are, they're certainly early in the run, they may have them, but at a much lower level, and they're amplified as the summer goes on. And for example, that Ninophytes worm, it's not going to multiply in the fish, so they just keep getting reinfected and reinfected. We don't have an adequate sample from ocean caught. That's why we're hoping that that the sports fishermen group, you know, we don't have enough ocean caught fish at all to say about the gut disease in these fish. Are we starting to see enterocytozoan before we see the gut disease? So long story, a simple question is we just do not have good enough data on the ocean fish or in the fish right after they come back. You know, like in late winter, we see it as early as June, but there's still a gap between late winter and June and early in the run, like it's still in the Columbia before they get to Willamette. We just don't have samples yet on that.
So hello, I'm Franca Hernandez.
Hi, Franca.
My question is, are there experiments or other areas in the world that are doing what you're doing, like Russia or the Scandinavian countries?
Not that I'm aware of. We have had some people from raising Chinook salmon in New Zealand in net pens that heard our talks, and contacted us, and we looked at some of the material. They have an enteritis disease, but it's something different than what we're looking at. It's a different pattern of lesions. I don't know if anybody else is working on these. What I'm finding is, and we're trying to, prespawn mortality is not a high priority. I'm really glad that Corbin was able to get funding for this. An attitude that we often get is, well, we had 67% prespawn mortality. That means we still have 33% fish, and that gives us enough eggs to run our hatchery program for the next generation. So it is really not a high priority for most funding agencies. It hasn't.
I have a.
Maybe we'll change. Yeah.
Go ahead, Vicki.
Hi, this is Vicki. I have a question for Dr. Schuster. Because there's a long history of oral tradition of salmon with the tribals, are there any stories of previous large diebacks in history that might have been caused by other pathogens or other conditions? That other times, not just modern.
Yeah, there certainly are. The one that immediately comes to mind is a story of overharvesting. And so there was a period, learning period, I would say. Overharvesting, a period of when the sort of, I guess, the management practices were starting to become really pronounced. And so there was essentially too much harvest, and it's called the story of the lost salmon. At least it's a legend that's consistently passed down through the Warm Springs culture. But they talk about how the river was depleted of the fish resource even prior to colonization. So there was even then a period of this idea of overharvesting and what that sort of means on a management level for the ecosystem.
So I have another question for Dr. Schuster, and that is, I noticed the interested parties that were involved in, that you'd drawn into your research work and kind of coalition didn't include, you referred to them as, stakeholders. There's shareholders. There's like the utilities that are at cause, or the Corps of Engineers, or the Pacific Ocean Energy Trust is trying to develop marine sanctuaries, but their infrastructure is understudied as far as its impact. Well, what's the history of those groups engaging, or is there a reason they don't?
Okay, I think there's no reason we excluded any of the groups specifically. I will say to engage with some of our other shareholders, like the Columbia River Intertribal Fish Commission, some of our tribal partnerships. They have had a history with some of those utilities, specifically Bonneville Power Administration. And so with that, it was sort of trying to carefully navigate that sort of that realm as well on our end, to include some of these perspectives that have historically been disregarded.
I can add to that. So one thing is, I've not followed the politics on it. Army Corps of Engineers provided a whole lot of our support early on for our research, but that tried, for whatever reason, I don't know why, that is not, they're not providing funding now.
I'm Joel. Excuse me. Historically, I wonder if there's been any change in the size or the weight of fish because of maybe these factors. Because you see pictures and photographs of earlier, fisheries, and they're these huge fish, and it seems like they're smaller these days. I don't know. Is that a factor at all?
I can answer from a fisher's perspective. I would definitely agree with that. My grandparents have, pictures of fish that are as tall as them, and we're pulling out fish that are, you know, half the size of our arm. So, I mean, I do think we have seen a decline, and I think that's specifically true, and I think it's even founded in the scientific literature as you move up the Columbia River, as these fish have to go beyond multiple dams and barriers to reach their ancestral spawning grounds. I know the sturgeon, for example, are a good species where we see, like up towards Colville, those upper regions of the Columbia River, extreme decrease in size from what we would see down here towards the mouth.
Yeah, it's an interesting question. When I worked for Fisheries and Oceans Canada from 1988 to 1999, and I noticed, I don't know the cause, but like there was a big sports, draw up the Quadra Island of Vancouver Island for Tyee salmon, these gigantic salmon like your grandfather, and those were going down. And one thought was that if you're because the fish are going to come back 3, 4, and 5 years, you're taking you're spawning more of the 3-year-old fish and not enough of the 5-year-old fish. And two things that, so there's pressure. I'm just outside of my area. I'm not the fishery biologist might just say you're full of it. Is that one is you're selecting those big fish, like catching the sports fishermen. This is up on. You're taking, everyone wants to catch a 45-pound salmon, so you're selectively moving those big fish. And then, like you say, I don't know what the challenges of the new challenges in survival to spawning. Maybe I'm just throwing this out. Maybe they favor smaller fish. And that could be why these upper reaches, why particularly the further you go up, the more challenging, the fewer big fish that you have. I don't know. That's something that's a good question. I'll talk to my fisheries colleagues. That's a very good question, the answer. I'm intrigued by what that answer is. Because that's a real phenomenon. What the cause is, I don't know.
David again. So you were saying 33% of the fish make it to producing eggs, right? So do you see any signs of these various diseases in the eggs or in the new spawn?
No, we haven't documented that at all. I mean, we don't have the data to say, okay, these populations that are being pressured by pre-spawn mortality, how is that affecting their progeny? Other than it's intuitive, it's like you're going to have a smaller gene pool. I guess someone could argue and say, well, we've really tortured those adult fish and only 10% of them made it, so those are the toughest 10%, so maybe that's good that we do that. I'm not advocating that, but I can imagine somebody saying something like that. You're basically culling out the weak ones by having them die from pre-spawn mortality. It is an interesting phenomenon. I talked to Dr. Peterson more about this. It's pretty clear that the higher numbers of fish there are in the river, the higher the pre-spawn mortality is. So then we start thinking of infectious. And he always asks me, Mike, he goes, Mike, what do you think is causing it? Could be, well, it could be disease transmission and stuff could be affecting it. So.
I can see it again. Sorry. We have the Santiam River just right outside the door here, and we have Detroit Reservoir, which is going through a drawdown. Have you seen anything happening on the Santiam at all that pertains to any of this?
Well, we see the disease in the Santiam stock.
Okay.
I don't have any information on pre-spawn mortality off the top of my head or that, but we have seen it in the South Santiam stock.
And dam drawdown? No?
I don't know that much about that.
Okay.
Hi, my name is John. Are there any selective pressures that are in effect? Meaning, if this PSM is causing a decline, I mean, the ones that make it through, are they more immune to getting it, and would they pass it on to the new fry, and so the next generation would be less susceptible to getting PSM?
That would be a hypothesis, and I can't really answer that. I'm looking, this guy, a group from Idaho, when I first was working on this stuff, they had some models, and talking about in wild stockfish, that they predicted that when pre-spawn mortality started getting greater than 60%, that there was a correlation of decline in survival of the stocks. So that's not really answering your question, but I wish I could find that slide. I couldn't have been asking my colleagues the data. They run the models. So basically, pre-spawn mortality is a normal phenomenon in salmonids, not 80 or 90%. You know, it's going to be, and it's going to change from year to year, but it's extreme now. Well, when you have 100% pre-spawn mortality, there's the end of your stock. You know, so that the whole year is gone if you're going to play that game. You know, so.
This will be the last question.
Hi, I'm Tom. Question for either of you or both of you, and that is, based on the data you've shared with us today, do you have any recommendations to make for fisheries management?
Okay. So the one recommendation, and Carl Schreck put that forward based on the study of holding those fish in cold water, is that you're not going to chill the Willamette River. That if you wanted to, you could, if someone wanted to make the investment, you could do like they do at Minter Creek, is take the fish and put them in tanks on cold water through the summer to get them to stop it. I'm not recommending. That's a method. Someone said, "What can you do about this?" Figure out a way to put the fish in cold water, and they will still sexually. They're happy to be at 12 or 14 degrees Celsius. So it's not like, oh, they're going to delay their sexual development. No, they're fine. You're going to get lots of, they'll still mature in September if you could figure out a way to chill the water.